Hi everyone, my name is Rodrigo Zanandrea. I'm a Master of Health Science and I will show you my work entitled A Transitory Changes the Behavior and Causes Neurochemical Impairment in Adult Zebrafish. The methionine is an essential amino acid important in many biological functions, like methylation process, and act as an initiator codon and participate in the synthesis of other amino acids. However, increased levels of this amino acid in plasma above of 40 micromolar per liter indicate a condition known as hypermethionemia. This pathological condition is caused by genetic causes that entail in deficiency of enzymes participating in the methionine cycle, or non-genetic causes, such as inappropriate diets, liver disease, or premature birth. Toxicological alterations of a persistent hypermethionemia include myopathies, neurological dysfunctions, and liver disease. All these well documented, but little is known about the effects of a transient plasmatic increase of this amino acid. Therefore, the objective of this study was to evaluate behavioral parameters, oxidative profile, and neurochemical analysis in adult zebrafish during transient exposure to meth. So, adult zebrafish were randomly assigned in two groups: the methionine group exposure to 3 micromolar of methionine for 7 days, or the control group exposure only to water. After exposure, both groups were maintained for more 7 days in aquariums containing only water. Then, the animals were submitted to the novel tank test, social interaction test, mirror-induced aggression test, and inhibitory avoidance test. The brains were collected to evaluation of oxidative parameters and analysis of amino acids and neurotransmitters. Our results showed that the exposure to methionine caused a decrease in the distance moved and velocity, whereas increased the time in the upper zone. The methionine group showed decreased time in the mirror zone, indicating reduced aggressive behavior, whereas the social interaction did not change. Exposure to methionine caused memory impairment. In the oxidative parameters, the catalase and superoxide dismutase activities decreased in the methionine group, as well as the content of total thiols. Besides that, the transient exposure to methionine caused a decrease in epinephrine and cysteine levels, and increased carnitine and creatine levels. Our findings indicated that the methionine transient exposure causes behavioral changes such as decreased anxiety, decreased aggression, and impaired memory. Also, the mechanisms underlying these effects are related to changes in the antioxidant system, amino acid, and neurotransmitter levels. Therefore, individuals with diagnosed hypermethionemia, dietary supplements without a professional orientation, premature babies, or patients with liver disease deserve special attention. I'm very thankful for financial support of CAPES and CNPq and of my laboratory of neurochemical and psychopharmacology. The all work is published in Molecular Neurobiology. Who is interested, search for withdrawal effects following methionine exposure in adult zebrafish. Thanks.